Listen on iConversation med
Charlie Rignaia.
Ann-Cathrin and Bo Engwall von Schéele.
Schéele.
Welcome to the podcast. Nice to see you again, Ann-Cathrin.
Nice to see you too.
Nice to meet you.
Yeah, nice to meet you, Bo. And congratulations on being a Nobel.
Yes, nowadays I am. And it's due to Ann-Cathrin's intervention, I must say. I'm too lazy to adjust to such things. But yes, I will present myself. And I have my education in mathematics, you might say, because I've been studying at Kungliga Tekniska Högskolan in Sweden.
Royal Academy of Technology.
Oh, maybe it's called like that.
Yeah.
Yeah, and I must boast a little bit. I have the highest mark in mathematics in the 5 first mathematical courses. Yes, you might say, and with the mark also, the final mark. But then I skipped studying because I had become a long distance runner, and I was very serious at that. And so I was also competing in international level in the Swedish team of marathoners. So running has been ever my main focus, and also stock dealing and analyzing, but that's for my living. And but I'm also very interested in immunology, since my wife has she was actually a teacher for me for some time when the swine flu was and the swine flu mass vaccination was going on, would be going on in 2009. Yes, and so I'm generally interested in these kind of sciences, you might say.
Yes, and also we can tell that we analyzed biotech industry projects for investors, because I had the technology background. I can go into the projects. I could help with the understanding if it was a good project or not. And Bos was calculating and looking for the numbers, so to speak. So
and also I've been analyzing the research also, because I can do that if I just get the main education in the topic.
Mm-hmm.
And what about you, Ann-Cathrin? In order to make this a standalone episode,
you
presented
I'm not
yourself
going to take as long as I did before, but generally I am the reason why I'm interested in immunology, because I had a disease when I was young, 11 years old. I was at the hospital for 1 month, and I couldn't go to school for half a year because I had meningitis in the brain, so I couldn't walk properly and so on. And that time it was very boring for a young child. So I had a lot of thinking about why I was so ill and how I become well and how it works and so on. So that actually occupied my mind for a long time. And then when studying, most of my family have not medical education. They are economists and engineers and technology educated. So, but I was sort of interested in medicine because I had that experience when I was a young child. So I decided to go to the pharmaceutical education because I didn't like, I was afraid of blood and cutting in people and things like that you have to do when you study medicine. So I choose to study pharmacology or pharmaceut, because that is also how to heal people, the medications and so on. So I was interested in that. But during my education, I actually came in contact with gene technology. And that was new in this time, in the end of the 80s. It's a very boom to study gene technology. So after I was finished my first education, I decided to go on to studying gene technology. And there was a education called biotechnology, sort of Master of Science education. So I went to that, and during these courses, I came in contact with immunology, and I was immediately hooked on that because it was very interesting, and it actually associated with my former experience as a child. So I asked one of the lecturers if I could come and become a PhD student. And so it went from there. And I was studying the MHC complex. And the MHC complex is actually molecules that present all the foreign things to the immune system and decides which route the immune system should go, because immune system is mostly cell-cell contacted through this MHC complex with foreign peptides from viruses or bacteria, and from that it activates a lot of chemical signals like cytokines that will tell the immune system which way to go to conquer this infection. And that was very interesting. It's very complex, and I like that. And it's very general because you get to understand how the immune system works in a more level that is higher. It's not so specific. It is specific, but on the same way you understand the immune system, how it works quite well. But during that time when I was studying genes of the MHC, I saw that approximately 23% of the area contained a lot of repetitive sequences that were very similar to virus. So they were virus related, and of different sorts. Is called transposon, retrotransposon, endogenous retroviruses, etc. And I wonder, why do we have almost a fourth of our genome with this kind of sequences? It's kind of strange. People said in those days, oh, it's junk, it's nothing important. But I could see that it actually influenced the development of the immune system. It has actually led to double of genes, like take away of part of genes, inactivation of genes in this area and so on. So it had a very significant impact on the development of the immune system. And therefore I became very, very interested in virology. So I started to study virology, and I found a man called Temin, who won the Nobel Prize. He actually isolated leukemia virus causing cancer, and he actually suggested that virus could have been developed from genomes, from these
Human.
I know all kind of genomes, and when I got this idea from this man, I started to look at my data and say, yeah, sure, I can understand that they are more primitive. So I actually wrote together with my colleagues. We wrote an article to review article about this, and the theories that viruses could have been developed from these elements, and that it was a part of the evolutionary path of you. So it's easier to make evolution if you have this type of elements, because if you have a virus infection or something like that, it could stimulate these elements and rearrange the genome in certain ways. That could be beneficial or bad for the individual. If it's bad, you have cancer. If it's good, you will have development. And actually, there are some examples of this kind of development. For example, that primates can produce lysozyme in the mouth, in the saliva, and break down food already in the mouth. It's typical for primates, and actually that is been generated by such an event. So it can be good, but it can also be bad. So, but it makes things, when you change your environment, it actually can go very fast. If you have this kind of mutation everywhere, there will always be one that will be beneficial and that will also lead to new species, for example. And I could see that these events took place when primate species were divided. Anyway, after that, this article and when I did my dissertation in Uppsala, I had an opponent from the Paul-Ehrlich Institute in Germany, and he actually invited me as a postdoc to come to Germany to work at this prestigious institute. And actually, at that time, the institute was controlling the whole area of protecting the German people for infections and things like that, similar to the Smittskyddsinstitutet in Sweden. And they had virus science about viruses. They had a lot of virus immune system co-development studies, which I actually was involved in. But the main thing is I learned a lot how you should think in these cases when it comes an infection how to proceed in a population interest, or what kind of factors that you should think about for the population, and that was very interesting for me because I got a more broad picture of how to work in these situations. And the head of that department was Reinhard Kurt, who was responsible for decades for this type of threats to the people. Anyway, after that, my mother got ill, so I went back to Sweden and ended up working at the biotechnology industry and working with the design and development of vaccine. And as a head of that department, it was very interesting, and I learned a lot about how to produce in cells and big scale production things. It's different than doing it in the small scale. So it was very interesting to see how things work and what the regulations are and so on. So I learned a lot about vaccine safety, what kind of factors are important and so on for the authorities that we have to test and so on. So that was also very interesting. And I think it was 2006 or something like that, I discovered that when they were doing their clinical test, there's a lot of things going on, so I didn't really like to continue this path because it made me feel bad. I didn't like it. So
they were cheating.
Yeah, they were doing. But
can
we can talk about that
on,
later on.
Can you elaborate a little bit on that right now?
Yeah, sure. I mean, I actually first it was very secretive. Nobody that not working directly with this got any information, which I think is very suspicious if you are serious. Why don't discuss what your plans are and so on? But no, it was very secretive and whispering around in the corridors and so on. And then at first I thought that was strange, but I focused on my own responsibilities. Later on, when they showed some results, I noticed they have compared total different things with each other that made a false picture of something that's very effective. But it was actually different scale and different, which you shouldn't do in science. You should have the same level, so to speak, to be able to compare correctly. You should not have different scales on things. And another thing I noticed when they reported from the side effects, I saw pictures of a guy with a very big arm. It looks like he had edema, I think it's called in English. I don't know, ödem på svenska. Anyway, it was very, very big, and they described it as a small swelling at the injection site, which I found was very wrong. This guy really had a problem, and it was not something that is just a minor effect. So I didn't like that, actually. So I decided I have to leave, and they tried to force me to stay with different kind of
Threats.
And it was a very tough period because I was a senior person in that company. I had 6 months leave, so they had to, and they actually said, oh, if I leave, I can't look for another job because I've learned things at this place that I could use, and I could not. They have to admit to if I got a new job, if it is okay, so I will not tell secrets from the company and use them for my own benefits, and I should have to pay millions and millions as a damage to the company and blah blah blah. And I felt very bad in that situation, so I contacted the union, and they helped me to start with it. They never actually met someone who was not allowed to leave their job. And to be honest with you, you don't feel really motivated to work when when they treat you like that. So, but I managed to help with the help of my husband to get out of that contract.
And we started a war against them, and that was very funny, I think.
In what way?
Well, we did sank the whole company, and also the tax reduction, or they were going to use the losses of about 500 million Swedish crowns. It was a huge, they had lost a huge sum, I think, and they were going to use that probably illegally with a big, well-known construction company, Peab, in Sweden, and I contacted the authorities about this, and the police, etc. And there was some interesting stories about that. There was a 3-article long in the internet news.
Realtid.
Yes, realtid, real times. But they are deleted now. I'm sorry about all these interesting articles. But well, they ended up in as a pole in the Fyrisån.
No, actually, it was like that. I had 2 shares, which my husband said I should get insight into what was going on. And with these 2 shares, they couldn't go through with this. I don't know how skalbolag in English. I don't know.
Yeah. Well, it was.
They wanted to empty the company and just leave the debts.
The tax reduction.
And that was going to use by the construction company to reduce their tax. And from that, they should share this with the pharmaceutical company that was developing the vaccine, so they could continue. And I didn't want that to continue because it was unethical.
Yeah, I can also explain. The company was called Resistencia, and they constructed a holding company, Resistencia Holding, which was going to receive this tax reduction, the part of it, about 30 million crowns, and they were going to continue with a very bad new chief executive officer, who was Ann-Cathrin's boss. He was the overall chief for the
science, but
yes.
he actually couldn't.
He was totally incompetent, I've heard.
He didn't know anything about what we were doing. He just, was there as some kind of, he was strategical and just collecting information, but he couldn't really.
Actually, I must add that one of these chairs was supposed to be mine, so we were going to own one share each, and we stopped that. Well, finally they got our 2 shares, but then the whole tax reduction plan was synced to the bottom of the sea in the Fyristan. It was a big article about this in Uppsala Nya Tidning, Swedish paper local in Uppsala, and it was published in 2009, and so that article also is very difficult to reach because everything is deleted from internet by these crooks. So
but anyway, they didn't get any money, so.
Can I just bring in a side note there as well, because it's very interesting that you say that the articles and the information was deleted from the website, because I noticed that as well. When
they were so-called caged. In those days, at least, you could when you Google for some article, there were also caged versions of this. There was maybe, I think it was 7 of these at that time around, and they managed to delete them all. But of course, we have the original articles. Journalists who are interested in getting those, they can have them from me, for example, and in PDF files. And I know there is one other journalist who has, Per Shapiro, has got these, and he was looking at them before he did some interviews with Ann-Cathrin as well. So it's the chief executive officer at that time, it was one of Anders Wall's boys, you might say. It was Marcus Bosson, and I always call him Blossom, but it's an association. But he was actually maneuvered out of that Resistencia company from your chief. So it was intrigues also within that company.
But that actually describes how things work, because all the people involved in the strategic were former people that had high positions in AstraZeneca or Pharmacia, big pharma companies, and that shows that how it actually, I think, shows how things work generally. Normally, someone like me would not come so well in contact with these people if I work in a bigger company. But because it was a smaller company, we could see this. I could see this in front of my eyes, and that actually made me realize I don't want to work with this anymore. Even though I care about people who need this or whatever, I think I didn't feel motivated to put my energy into something that is so corrupt. So that's how I felt, and I am happy that I left there, because it made headaches. It made me feel headache and really bad situation.
I think Ann-Cathrin she has developed in an informer, you might say, and educating all those who are reading her articles at Facebook, and so she has done something better now, I think. And she agrees.
I actually feel much better because I'm helping people now. In that situation, I didn't help people. I just helped these people who were should make money on what I was doing, and I didn't like that, because it was not for the benefit of the people. And I actually have, because I was ill myself as a child, this is why this is my motivation. I want to help people to get better, not you know.
But nowadays we are also contacting the authorities in Sweden, and we are acting and learn. We are trying to learn them how things should be regarding the quality of the personnel there. For example, the Swedish authority, Folkhälsomyndigheten, they had before they had a incompetent epidemiolog.
Chief epidemiologist.
He was incompetent, and we prayed that he had to resign because of the competence, and also because he got money from several vaccine companies and several well-known, like AstraZeneca, etc.
Was that Magnus Gisslén, or was it another?
Yeah,
it was Magnus. Yeah.
Yes. And we demanded he must resign, and we did that very with good motivations. And about 1 or 2 months later, he was off. He
resigned.
Yeah, and they are corrupt also. They always have the chance to resign themselves and say.
But that is normal.
He actually has written about this on, for example, LinkedIn, and he has some good points. He has said that they are incompetent in the authority, and he's correct. So he was not totally wrong. The problem was that he himself was incompetent.
But the problem is that the people who are making the decisions are not really scientists or people who are real experts. It's mostly political demands from that rules how to act in a situation. Not like I was taught in Germany, because what I was taught in Germany is what I now suggest. Things that we knew 20 years ago was true. Like, for example, you should not vaccinate more than 10% maximum to protect yourself against the virus, like influenza, because it mutates so fast. So the vaccine becomes ineffective much faster, and the people who need to be protected lose in that situation. So it's not a good idea, and that was the first thing I was reacting against in 2009, because I thought this is really not the way to do this.
Yes, I would like to add, I forgot something rather important, I think. About 4 months after we had started with this, you might say, research and intervention regarding the swine flu pandemic and the mass vaccination, I got published in British Medical Journal. It was a long, so formally rapid response, and it was you might say a development of the first analyzing that Ann-Cathrin had done in 2009, just we started them 4 months earlier in just
summer 2009.
Yeah, it was about
August, maybe.
It was late August, late July or early August that I had actually asked Ann-Cathrin, should we vaccinate us against this new, and she deduced just on we call it 15 minutes, and she deduced no, we shouldn't, because and then she explained all things about antibodies and the T-cell immunity, which is more important
viruses.
In interaction with human interaction with viruses. And but from that, and her first article in Svenska Dagbladet, it was the 28th of August that year, she warned against doing this because it was not optimal to mass vaccinate all. You should only, if the vaccine is good, you should only vaccine those, a little minority that has actually a positive cost-benefit ratio, or negative, you might say, more benefit than negative effects.
So what I noticed is that they wanted to start to vaccinate small children. And for me, this is wrong, because normally before puberty, you have a better, you don't have, your immune system is not that strong, because the sexual hormones is not activated in the same way. So that's actually, your immune system is not fully developed, and that means you don't get so sick when you get your first flu when you are a child, and that can protect you for the whole life with a T-cell immunity. And what my husband and I saw is actually that when we looked at the infected people who had the age that they had gone through the flus, Asiaten and Hong Kong flus, had better. They didn't get sick from the new.
Actually, it was the most non-severe pandemic ever, probably, because there were only about 15 people in Sweden who died from the swine flu. And normally they die about several hundreds in Sweden each year from the
normal,
the seasonal flu. So it was they were extremely good protective at from this new, as they call it, the new influenza, and they were daring stories about this how sick they were going to be, and but they were totally incompetent. They didn't know anything, obviously, about the interaction between human beings and viruses. They come from the bacteria.
Bacterial science, and that's different.
Why did they do that?
I don't know, because Smittskyddsinstitutet, that was before Folkhälsomyndigheten, actually was a lot about bacteriology. So many of them were bacteriologists, and they moved along with the move to Folkhälsomyndigheten.
Yeah, they thought it was the same type of interaction.
No, but I think that they have this education, and I mean, anyway,
they were incompetent.
Yeah, but what I was going to say is it's the difference between the T-cell immunity and the B-cell immunity. B-cell immunity is antibodies. T-cell immunity is when you get a virus infection, something goes into the cell, and the cell expresses these new viruses, and it's mounted in the cell. And at that time, the viruses also express some viral protein on the outside connected to this MHC that I was talking about earlier. And this is actually recognized by the T cells that it is foreign. And these cells are killed they give them a death signal, and the cell do apoptosis, kills itself. And that is the way how you get rid of an viral infection, that the cells producing the virus are killed, and antibodies cannot do that. What they do, they can stop viruses outside the cell because they can bind to the outside of the virus. But when the virus gets into the cell, all the proteins you have an immunity against, because all the proteins are produced there and exposed on the infected cells. And that means you have a memory of T killer cells that can act upon infected cells. And these inner proteins that is inside the particle that is hidden from the antibody response, it don't change that much because it doesn't need to, because it can still infect. So what is mutated is often the H and N and the influenza, or the spike protein of the coronavirus on the surface. Exactly. So the inner protein are more conserved. So when you get a T cell immunity, you get immunity against the whole protein, even things that doesn't expose to the antibodies. But you will get an infection, but it will become much, much milder. Yeah.
And I want to add that it was this that Ann-Cathrin told me in 15 minutes that time in late July or August, beginning of August 2009. And I immediately understood all these things. And therefore I said, you must write an article about this. And that was what happened. She did that. A few weeks later, it was ready, and it was published in Svenska Dagbladet, and it was a big discussion there. It was about 300 comments or something like that.
Yeah, it was the most read article that week in Svenska Dagbladet.
Yeah.
And just to be clear, was that during the swine flu?
Yeah.
2009.
Yes.
And it was published 28th of August that year. And about that time also, I had started to communicate with Annika Linde, who was the responsible chief epidemiologist at the Smittskyddsinstitutet Institute.
The old.
Yeah, at that time. And I started to claim that this is totally wrong, as Ann-Cathrin has said. And it was a correspondence about 2 or 3 times, and then she gave up. She didn't want to hear about this. They don't want to hear the truth.
Yeah.
That's not what they like, because they had planned so much to do these bad things.
And I think they didn't understand. They thought it should be good, because they thought they should stop to stop.
They were incompetent. That's what they were incompetent.
Do you guys really think they were incompetent, or was there just
No, they were incompetent regarding this, because I noticed that from her answers. But when she got my
argumentation,
then she understood that it was something, and then she didn't want to hear more.
But she didn't get the vaccine herself, so that is maybe an indication she understood something. I don't know. Anyway, I think that was when we were discussing the side effects on the TV program in Gothenburg in SVT, Swedish television, with the general secretary of Smittskyddsinstitutet and some other scientists that thought that this mass vaccination was a good idea, and they were discussing these 400 children or more that had received narcolepsy from the injections. After this discussion in the television, we stayed at the hotel, and I discussed with Johan Karlsson, who was the general director of the responsible for this decisions. He actually said to me, but Ann-Cathrin, I know maybe it was not a good this way and so on, but it was a good logistic experiment. And I looked at him and I said, logistic experiments, that is actually how to transport the vaccines here and there. This is not a good price to 400 children to get very severe disease. I don't think so.
It destroyed their lives.
Not destroyed, but at least it made their life more difficult. And it's not a nice disease. And 400 people is not so much, but it's young people who will suffer for a long time. And I don't think that was a really good outcome for testing the logistics.
You have done some research also in narcolepsy.
Yeah, I have done that, and so I know it's a very, very horrible disease.
And the thing about all of this is as well, what was the response from the media at the time when you published these articles? Because I was only, like, as I told Ann-Cathrin last time we discussed this, I was 7 years old during the swine flu pandemic, so I have no background in that. But during the COVID pandemic,
I can say something there.
Yeah.
Okay.
Yeah, I just wanted to tell you that I took 3 COVID shots because I work in healthcare, and I thought that the main they told us that it was safe and effective, and I wasn't more educated in the topic than that. So I thought, okay, I will take it so I don't spread it to the elders I'm working with. So there was no nuance in media that the vaccine could have side effects.
In those days, I was allowed, I was invited to television many times in Göteborg. I was invited to Aschberg's studio in Stockholm.
And interviewed in Expression, for example, and other media.
Yeah, other media, and they referred to it was not so tough in those days, but I was more like invited to be an example, how to say, I was often alone against a lot of experts and a lot of people who had, normal people who have ideas that was not scientific, and then it was me. So I was pretty alone against all these
you were already then an anti-vaxxer.
Yeah, in a way, I mean. But they said to me, when I was in television first time, after my article, I was sitting down with a journalist and said, you cannot be too scientific, but you have to play a role here. You have to play a role to be against vaccine or not. I mean, I said,
it was a TV show.
Yeah, it has to be interesting. And I said, what the fuck? I don't want to do this. I want to be myself. I want to be honest. What is this? So I actually reacted on the way they tried to prep me to do something that I didn't really want to, but I was well prepared, so I actually was putting them on place.
I want to add also my BMG article, then it was published 1st December of 2009, and it can still be read. And at that time I concluded much more, and for example, that you get the protection from if there were to be a new fågelinfluenza,
bird flu.
The bird flu, which was very much talked about, and how dangerous it was. You actually get the protection from bird flu as well, and that is due to these conserved inner proteins, and there have been studies about this.
Doherty.
Yeah, Peter Doherty from New Zealand. He was a Nobel Prize winner in
1996.
Yeah, and he was actually holding his speech in Uppsala University in 1996 as well. And
the same day as I was holding my dissertation, so I had to move my dissertation to the afternoon because he was coming there.
Yeah,
all the opponents and the people wanted to listen to this.
And he had done several experiments on mice and etc. in 2 years, all around these years, 2000, 2001, etc. He'd done several experiments about how protection from a seasonal flu, it actually, it was like this. For example, one study I remember, it was, they were immune naive mice that had, if they were immunized, yes, with the seasonal flu, and then they were protected. 90% were surviving from a dangerous for these mice, otherwise bird flu. And because only without this immunization before, they died 9 out of 10, I think. And so
they were protected by first being exposed to seasonal flu from the bird flu.
And that must have been, since the surface proteins were totally different, then it must have been due to the other type of immunity. And of course, that was the T-cell immunity from inner conserved proteins. And at the same time, it was published in about when the Swedish mass vaccination was starting. At that time, it was published in an article about these inner proteins, and they were actually concerned. It was proved between the different types of. In that case, I think it was proved between seasonal flus and the swine flu, because so they explained why there were protection against, and they also compared with the Hong Kong 40 years earlier, and there was similar type of protection and conserved the number of the percentage of conserved inner proteins were about maybe 60 or 70%. So they were actually very, very similar these different type of viruses 40 years later. So therefore this is a very important
factor, and I think that
and they didn't understand anything in the Swedish about this.
And that what is
Swedish
really
authorities.
practical sense is that if you have had one flu in your life, influenza, then you are protected against
rather well
almost all flus, not everyone, but almost all flu. But you can get infected, but you will not be so severely infecting as if you would have it for the first time.
And that is very important to state, that it's not now, it's the same thing with COVID-19 and the mRNA vaccine. It doesn't protect, it doesn't make if you are infected, it's not the vaccine that you get protected from. It's from, if you have, as nearly everyone who now are getting the booster, they have been infected before, and they have this broad protection from earlier, not only the COVID-19 for earlier COVID-19 infections, but also from corona
virus infection. That is normal flu infection.
Yeah, I mean common cold.
Common cold.
And that was also more or less proved as early as 2020, when there was it was this
boat.
Yeah, this cruise.
Diamond Princess.
Yes.
Diamond Princess.
Yeah.
Boat, a boat that transported a lot of elderly on cruises in the Asia.
And they were isolated. They couldn't enter
the vessel.
They couldn't go to land and leave the ship. And they were taking a lot of tests, and different studies, at least two, showed already then that about 25 to 50% different results of these elderly people that were infected by COVID-19, did actually have without symptoms. So, and of course it was due to this. Ann-Cathrin said that she speculated, and not speculated, she did use that they must have been protected before, because she knew about these corona colds. And just a few months later in the summer, they were published several studies, and they were not, of course, they were not talked about in media. There was a lockdown, not only people who were locked in the houses in different countries, they locked out the information and studies from about the autumn that year. Then when the mass vaccination was going to start, there was more or less in the whole world, it was coordinated, must have been. You mustn't talk about these things, about T-cell immunity, etc., etc. So it was a cover-up from the start, because maybe they thought it was to be good, this mass vaccination.
I think that they thought if people are not infected, they should protect them with this vaccine, the COVID vaccines. And I don't think it works like that, because even though you try to shut down the infection still spreads, the virus still spreads, but in a lower rate. But every person that it spreads to, the more effective the virus gets. So it's actually better that more people get the infection early in the process than later, because later in the development, the virus is more effective in infection and in fast multiplying, so you will get more severe infections. So if you lock people in, you risk more severe infection in a bigger part of the population, and that is not really good.
And that was also very interesting, because Sweden was an example of a different kind of intervention against the virus, since we, at that time, Anders Tegnell, who was also responsible for the mass vaccination and the buying of the pandemic's vaccine in 2009, he had learned something from his mistakes, and showed Sweden had a comparably low key intervention. The children could go to school, I think, and so forth, and it wasn't so no big thing, he thought, and that showed later that Sweden was the country with the lowest in Europe, together with they were, I think they, after around 2003 or something, they showed that Sweden had together with some small countries, these like San Marino, etc., that were protecting themselves, but they were the country with the lowest death rate, overall. The lowest overall death rate, I think, due to the over death rate. What do you think?
The over.
I mean, there were very low death rates in Sweden due to these and due to COVID-19 in Sweden on the whole.
During the whole period.
The elevation of death rate was low, among the lowest or the lowest in Sweden during all the pandemic's most severe times. So it was, and probably it was due to the early Swedish intervention that was without lockdown.
Because what you do then is that you will increase this T-cell immunity in the population, and that is what protects you from all the mutants and so on further on. And that means that even if you get new mutations that are more effective in infecting people and giving more severe infections, a bigger part of the population has protection, and that means that there's less virus in the environment that can cause severe diseases. So people will get lower amounts of virus when they are, contaminated by the virus.
So it's a myth now that it has been since 2022, I think, when one of the Swedish authority people were from the Folkhälsomyndigheten, often was cited in media, she said, but you will get protection against the severe COVID-19 and death. But that was a lie, or she didn't understand. They were incompetent. It's due to the broad T-cell immunity.
Yeah, and you don't get this broad T-cell immunity with the vaccine. They say you get T-cell immunity because the mRNA vaccine goes into the cell, but it only expresses the spike protein, which is outside. So
changes.
Yeah, this is the part of the virus that changed the most, so that it will be short-lived anyway, compared to if you get T-cell immunity from a natural infection. That will last for much longer time because you have a much more proteins that will be recognized, viral proteins that will be recognized by the immune system. So that will protect you, and they are more conserved. So it's probably can reduce the severity of a lot of coronavirus.
And another mistake regarding this, they also said people should get vaccinated after having, even if they had been.
That is crazy.
And yes,
that is really crazy.
So why is that crazy?
They only add the side effects of the vaccine.
Yeah.
So it's totally incompetent. So they must have been corrupt, since there were no immunology reason. They had some study, and they were also fake studies. They talked about super kind of.
Yeah, there should be some kind of combination immunology from the vaccine and the natural immunity, but that is wrong.
Yeah.
For what you are doing when you give more injections with something that is, normally when you get infected, you produce mutations in the spike protein, even though they are not spread. You produce a lot of viruses of different kinds, more or less. There will be mutations here and there, so you will have a much broader response also to the spike protein. But here you inject an mRNA that produces the same kind of spike protein in a lot of people, and that means that when you have had the infection with a broader repertoire of antibodies or so on, then you have something called affinity maturity of antibodies. That means that if you exposed many times, the antibodies will bind stronger to what you are exposed to. They become more effective. And if you then, after having this more broad response, then you focus on just one and old part of the spike protein that you get immunity against, then you will specify. So your immune system will go from being more broad to being specific on an old variant of the spike protein, which means that you will get a more not as good immune response.
And also there's a shift towards other types of antibodies. They call the IgG4, and they are actually used in other types of when you're allergics. They use this reaction.
Mechanism,
So actually, it's been shown in a whole American state. They showed that after the more times you had, when there were, if you had taken five vaccinations, the risk for getting
tolerance development.
And
not react.
They were infected the most. The more you had these vaccinations, the more risk for infection. And the ones who had the lowest risk of infection, it was the unvaccinated.
But I can explain. I think it's a little bit unclear.
Can I just ask you a question before you explain that? Because I have had this in my mind for a while now. I don't want to interrupt you guys, but I just want to be clear on this point. So you said a while back ago that if you have had any, if I interpret you correctly, if you have had any seasonal flu before the coronavirus, you automatically get protection from
not seasonal flu. If you had a coronavirus infection.
Okay.
It's a type of codes. There are corona codes. I think it was they say about maybe 1/3 of common codes are corona type. So
and there are already a lot of coronavirus circulating in the population.
And that's why Ann-Cathrin deduced that, and I realized that also in 2020, that was why the younger children were protected against it, because, or at least they were not sick. And that was because of the T-cell immunity. And also in their cases, because there are also conservation of some part of the spike protein from, it's as early as from 2003 when there was SARS-CoV-1. Some parts of the spike protein, I think it was the S2 part, it was conserved in and probably from the SARS-CoV-1 to corona codes, and then in the last transformation to the SARS-CoV-2. And it wasn't the S1 part. The S1 part is the part that is responsible for transportation for the infection.
Partly,
to the receptors.
Protein.
Yeah, but the S2 was a bit more conserved due to it's only responsible for the transformation of the virus into the cells.
Its structure so it can get into the cells, and that is a very important mechanism that probably if you change this doesn't work. So the virus will not be able to to get into the cell if that is too mutated.
But the S1 part is more important, since it wouldn't even be, you wouldn't be infected.
No.
If that.
Anyway, I wanted to explain what Bos explained before that was maybe a little bit difficult to understand. It's a process called tolerance, and this process tolerance is when you don't get the reaction from the immune system, and you need higher stimuli to get the reaction. And that is a way that you treat people who have allergies. You expose them to higher and higher dose of what they are allergic to. And that means that they will not react as strong for small doses of what they are allergic to. So that makes their life easier. Maybe not take away the allergy, but they need to have higher doses to react. And that will make their life easier. But if you expose yourself many, many times for a vaccine and you get tolerant to spike proteins, maybe not that good, because it can lead that you not react as fast as you need to if you are infected with the coronavirus. And that could be dangerous, actually. You can get more severe because you get higher levels of virus in your body and before your immune system acts, and that means that you will get more damage of your tissues, probably in the lung and so on, and that can lead to severe secondary effects like bacterial infections, sepsis, and other things, and very difficult infections. So if you are tolerant to a virus, it's not that good, and that actually was Bos explained. They saw after 5 doses that
also early they saw it already. It was a continuous process, you might say. So after 2 or 3 times, they saw it also that this type of IgG4 antibodies, they were actually dominating with, and
that's actually inhibit the immune system's response.
They don't inactivate the virus. They accept the virus.
They don't react against it. So that is quite not a good strategy.
We must also now, when we talk about also in COVID-19, my area, most interesting area about the cheating of the studies, because it's been continuing all the time, more or less. And the thing I discovered in Israel is important, very important. And it was their 3rd vaccination against COVID-19.
Booster.
I noticed in without knowing anything about that, I noticed that Israel in the autumn, there were huge, rapid deaths from COVID-19 in it was in the beginning of August, absolutely beginning of August, and they were rising from about 1 to 2 deaths per day, I think, around to just within a few weeks, about 40 deaths, and this was the second big death period, I think, from COVID-19 in Israel. And at that time, or maybe it was the third, I don't know exactly now, but at that time, I didn't know anything about why, so I Googled about it, and after some Googling, I understood that, they had started a mass vaccination in
campaign.
And it was at that time it was the high risk group of 60, all the 60 years and 60 plus, 60 years and older, and it was 500,000 of people who were vaccinated or got their first booster, you might say.
The third.
No, the first booster, it was the third.
And
the vaccination, and they got that. They received this booster during only 10 days in between. It was the 13th July to 8th of August. And all these people getting boosted. And at that time, also the death rate in Israel from the first day, you might say, you could see that it was a death rate from COVID-19 or with COVID-19 that was exceptional. And the thing is, I at the same time had during the summer noticed this type of reaction or these results in death rates and infection rate. They were going together as they usually do. You get infection before you eventually die, if you die. And I had noticed that in about 10 different low developed countries, the poor countries, during the summer and autumn, and at the same time, for example, Vietnam, it was nearly exactly the same time as Israel. But in Vietnam, it was the first
injection.
The first vaccination, and it was January. It was all these examples were about an initiated mass vaccination or elevated speed of the mass vaccination in time that you saw this. And it was, as in the Israel case, it was a momentaneous higher degree of the...
It was a high decrease in death rate.
Increase in death rate from the... So not only more infections, but and not more only deaths, but it was also a higher probability of dying if you got infected. And in the Israel case, in the total population at that time, I calculated from official sites like Worldometer, who had published these statistics, and I could see that it was the death rate was over 100% higher from infection if you got infection. And then I started to I started to do some calculations, and I found that it could be around maybe over 6 times higher death rates among actually the I mean, there were a lot of infections that were not causing death, and usually it's those, for example, the rest of those who were not boosters at that time, and that was a lot of people younger than 60 years. So they also got infected but didn't die. But I did my calculations and found that it should have been something like a 6 higher death rate about from those. And there also was a comparable group of in these
studies.
There were no studies involved in this case. It was my calculation. Okay. And I did some. I have all this I published all this on Facebook, and I also have it on my homepage. The exact calculation I did, and I found it was about 6 times higher death rate if they had got the booster compared with those in the similar ages not have got the boosters. And I also did use that overall there was let's see now what the interesting also about this was that this was, I mean, I've seen this before. I've seen it already in the Swedish swine flu mass vaccination in 2009. I immediately observed that there were very high in the same time as they started the mass vaccination. I postulated that there was an unbalancing effect due to the mass vaccination.
A lot of people got infected at the same time they got vaccinated.
And that was I then stipulated a theory, an unbalanced theory, and Ann-Cathrin agreed exactly with it when I deduced that since the pandemic's vaccine, it focused on producing antibodies against these
H and M.
Yeah, the outside proteins of the influenza virus.
Yeah.
And they used also for doing that, they also adjuvants, which in itself wasn't so good. but due to this vaccine pandemics, they got the immune system to focus on antibodies, and therefore it down-regulated the relative T cell response. And since many people had this before, a good T cell response, for example, from the Hong Kong flu or a seasonal flu, they had a higher risk of getting infected. So they actually promoted the outbreaks of swine flu during that time, according to this hypothesis. And but the same type of hypothesis could be
applied.
Yeah, applied to these, for example, the Israel case or the other types of 10 states.
Can I explain?
Yeah, okay.
What I feel is that if you have immune active, you stimulate the immune system in very high degree, like you do in vaccination. All the resources in the immune system.
Not all.
Now, most of the resources in the immune system will go towards to take care of that threat. And it's while if you get an infection in your
of a common cold type.
Yeah, a common cold type.
Like COVID.
It will not be handled until you have you will taken care of the vaccine. And that means that there's a possibility of the virus to duplicate and you have higher doses of virus before your immune system takes care of this threat because it's milder, because it's just in the lungs or in the airways. But so that means that you will have more virus in your body and when the immune system react. And that means that your damage will be more.
And the explanation for this, I think, my theory regarding this priority from the immune system might be that since, for example, if you got bitten by what tests they had in 100,000 years ago, maybe if they had a serious wound in the arm, for example, where they get the vaccine, then maybe the immune system prioritizes this wound, not the common cold you have at the same time. So it's very logical, and it's also what's also known in studies that this there is a balance between the antibody and the T cell response. It's called the Th1 contra Th2 balance. Th1 it stands for different kind of T helper cells, and the Th1 helper cells is those who are connected to the T cell response, and the Th2 are connected to the B cell response, which Ann-Cathrin has talked about.
Yeah, and that is with the chemical signals that it is different between the two.
And that is also very logical if you see it from an evolutionary state, since the body doesn't use these at the same time usually. If you have sufficient good antibodies, then you don't have to use the T cell system, because then they protect you. But if you don't, since they didn't have any vaccines at all at that time, but if you had that infection before with exactly the same virus, then you were protected from the antibodies. So it's very logical that the immune system has this balance between it doesn't it's from an evolutionary process that it's developed, and so why should it have a very big reserve for both? No, it's not optimal.
But you can say like this, the T cell response takes care of infections in the cells. It eliminates cancer cells that are dangerous to our body. It eliminates virus producing cells, while the antibody response, the B cell protection, it's normally taking care of things outside the cells, like virus particles outside the cell, bacteria that are outside the cells, etc., and like poisons and things like that also can be taken care of antibodies. So that is, if you want to sort of generalize it, it's easy to understand.
I should add that in this respect, that was you might say that all these discoveries I did noticed from these not so good developed poor countries, they were a confirmation of this unbalance effect. And when you have an infection starting or in low key, if you get vaccinated, then you elevate the risk for the infection to get more serious, and also for deaths and the probability of dying.
We should say also that our reasoning here is mainly actually for like airway viruses that infect your airways, that mutates very fast and spreads very fast.
That's basic.
So there are other diseases which you can prevent. You might prevent infection if you can start them before the virus is spread, and they are not mutating as fast. Then it's a different logic. But anyway, we should just mention that, so it's not related to all types of viruses, because you have to look at the virus, what type of virus is this. You have to look at what, if you have a vaccine, what type of design, because vaccines are not the same. It's not one vaccine, it works the same all the time. It's different mechanisms, and that you have to be aware of. And the third thing you have to be aware of is the individual. How is the individual? Is it what kind of, because you have individual ability due to your MAC genetics, that to protect yourself for different diseases. So we are different in that matter, and also we have different health background and so on that affect this. So these are the three main things you have to consider.
Yeah, and but now about the cheating in these.
Yeah.
There were 3 follow-up studies from this Israeli mass vaccination, and the first one I saw immediately because I was very interested in this case, and it was published 16th September 2021 in the autumn, and I was very very surprised that they said, oh, it was a success, etc., etc. And okay, I knew that it must have been a fake. I didn't believe in it, so I looked thoroughly on
the data.
Data and inclusion and exclusion
criteria.
What?
Criteria.
Yeah, the criteria for exclusion, etc. And I immediately noticed that they excluded the 11 days totally, the 11 days after each person's vaccination, the booster. They eliminated all the results. And
So
we know
crazy.
Yeah, they didn't. Okay, what happened then? If they died or so, okay, that wasn't so important. But they also, and they combined 2 very intelligent criteria to eliminate the catastrophe that was at that time. And they did that by also not following up deaths. In spite of the death rate in Israel went to the ceiling, and they only follow up severe COVID-19, and that was the smart trick. Before, in the booster group, there were so many deaths before these 11 days.
Yeah.
So they were actually eliminating a lot of these zero cases also at the same time. And that with these 2 criteria, the exclusion criteria and inclusion, or the definition of which type of effect they should follow up, and this severe effect only, they show that actually the number of severe cases would reduce from several hundreds to only 9 in the booster group. It was as we saw, but with so low probabilities, you understood immediately that it was a fake, and and that's how. And 5 days after that, it was published in British Medical Journal by my rapid response. It was very rapid. They understood because in British Medical Journal they were the only scientific, well-known,
more open-minded.
Yeah, more open, and they were actually very critical against the Pfizer phase 3 study, which was also full of bias, etc. Vaccine effect increasing bias, and also very bad in many respects, and terminated before, etc. So not following up sufficient time, etc. And so I got that published, and in just a few lines I described how they did this, and I think there were no comments at all about that, but it was very hot. The links to this article, it's always nowadays, if they can, in Facebook, it's inactivated links. If you don't get it, you can't just click on it and
it stopped.
Yeah.
But anyway, what is interesting is that I think it was just a day after, so they have changed your numbers in this article. Somehow someone has change his numbers?
Yeah. I mean, the number that was changed was, I stated that it was an overall increasement of over 100% of the death rate if you got infected in Israel at that time. And it was just the overall. And so I didn't, in that response, I had already calculated how high it should have been. And there is also, I must say about this, there's also from Netherlands, there's also one who, Dr. Simon, it was called. It was
scientist.
Yeah, he's a scientist, and he had published on the internet unofficially several diagram how these different type of groups, the non-vaccinated overall and these two groups of elderly, I think, that were boosters and not boosted. And these diagrams, I don't know how we had gotten these had gotten this probably from raw data in Israel, but I don't know how we did it. But there were some diagrams later that were congruent with my calculations.
Calculations.
Yeah, calculation. Yeah.
Calculations.
So therefore, it was independently, I think we two did this. And but I think I was the first who could realize that this was actually, that this type of unbalanced effect was
responsible.
Yeah, and it was the same type, that more or less same type of that in already discovered in 2009 when the Swedish mass vaccination against swine flu was initiated. So I had a very aha feeling that it was the same type, but since the general resistance, immunological resistance against the COVID-19 was less in Sweden, and it was also and in the COVID-19 pandemic, it was much more people that were infected, so it was a much severe effect. Actually, we have calculated that only this type of effect has caused an elevation of maybe several millions of deaths netto extra from the vaccination, and that's totally apart from the mRNA long-term effects from deaths from other types that we haven't discussed here yet. But we also know a lot about, and we theorized about it before the actual outcomes were published in many studies nowadays know that these
like cancer.
Yeah, and we didn't know before why there was a risk for this, and because Ann-Cathrin knew how this interaction could.
Yeah, I know the mechanism, how the immune system works, and from that you can predict a lot of things, because you know the mechanism for how both the vaccine work, how mRNA expressed, and also that.
What was the important difference between these mRNA vaccines compared to the earlier protein-based, for example?
Yeah, the difference is that this mRNA goes into the cells, and that it is stabilized, because you have like a fat around the mRNA, so to protect it and to deliver it through the membrane, because it's the same fat that you have in the cell membrane, so it just melt within the membrane and release the mRNA, and it's also very stabilized. Even though the T cells remove this sort of vaccine-producing cells that express the spike protein, there are cells that cannot be removed from the body by the let me speak. T cell, T killer cells. And that is the nerve cells, because if you eliminate the nerve cells...
Why do the vaccine reach the nerve cells and you get in the arm?
Yeah, it actually, it's difficult. It was transported, of course, through the whole body. But let me finish my discussion.
You haven't said that before.
Yeah. Anyway, it's the nerve cells, because if you eliminate the nerve cells, if you have an infection of a virus, for example, herpes virus, the protection will be more severe, because you can have mechanisms that doesn't function, like breathing or things like that, if your immune system attacks these nerves and kill them. And also it is the reproductive cells, because that's also very important for the next generation and the purpose of people to reproduce.
So the T cells doesn't?
No, they are protected. And also blood cells, because that is also the immune system. It's also protected, because these three systems is very important for the survival of humans. So that is why, but the other systems protecting these kind of cells, and that involves
oxidative stress.
Oxidative stress, and that produces a lot of aggressive components like peroxide, and the pH goes down, and that is something that is protective mechanism of the body, because your skin is low pH, your urea is low pH, etc., and that is to protect you from this kind of intruders like bacteria and so on. And when proteins come in contact with a low pH, they will denaturate. Actually, they will be inactive. They will lose their structure and will be insoluble. And if you have a lot of these products and also have a lot of inflammation, because oxidative stress and inflammation is a bad cycle, they activate each other. If you have a lot of inflammation and you have a lot of oxidative stress, you can affect the function of the nerve, you can affect the function of the reproductive cells. You can be sterile, for example, if you have a lot of inflammation and oxidative stress in your reproductive cells. Also, the nerves you can have nerve systems. You can have what do you call it? Like myeloid plaque. No, not myeloid plaque. Like Alzheimer, diseases like that. Amyloid plaques, sorry. You can get degenerative diseases, you can have problems with function of your organs and so on. So actually, this is the more difficult side effects that will affect those organs, like that are not eliminate cells. And if you eliminate a cell, for example, in the heart, that will not be
by T cells.
Yeah, by a T cell, for example. Then you not renew this cell, and you get scars.
And that's probably.
Can I just ask? Just ask a quick question. All these side effects could be side effects of the mRNA vaccine.
Yeah.
And that's always mostly the neurological she had talked about, but the most interesting, I think, importantly in the beginning was all these cancers that were for example, Ute Krüger, a Swedish former pathologist and etc.
She is a pathologist.
Yeah.
She was a former
but she had
head
to resign.
Of the department.
She couldn't. She was
She lost her position.
Yeah, she lost the and she couldn't be there because everyone was
Against her.
were attacking her. So it was very.
She saw a link. She actually started to notice a lot of.
Turbo.
Yeah, she noticed a lot of younger women had different type of cancers. Not only did they have very aggressive cancers.
Turbo cancer.
Yeah, very aggressive, fast-growing cancer. They had several different type of cancer, primary tumors at the same time, which is very unusual. So many tumors at the same time that are not metastasis, they are primary tumors, and they develop it in the same time in a very fast speed.
And how do you explain that?
She actually looked at the vaccination from these women and saw that all of them had the mRNA vaccine.
And
she made
why?
A link,
Why? Can you explain the mechanism?
Yeah, later. She actually wanted this to be more studied, and I think that's relevant.
Relevant. It's naturally.
She didn't
she had not seen anything like that. She was experienced.
She wanted that people should examine this. She didn't say this is a correlation, but I see this. We have to study if there is a correlation, and of course there is. Then she was like cancelled and so on, and
it was also a German pathologist, Burkhardt, who is now deceased, but he actually found in human autopsies in the heart, he found leukocytes that had attacking areas, and they had died from heart failure. So it was a very indicative research that he had done.
But actually, if you have damage of the heart, if you have losing cells, then the signaling in the heart doesn't work properly, and that could actually lead that you have a strange rhythm, and it doesn't work to start this heart rhythm, and that actually can cause heart failures, stop of the heart.
And this, this type of T-cell leukocyte is naturally the CD8 killer cells that are attacking the cells producing the spike protein. Since the mRNA has been found, reached that area, these cells, and that's the main principle that the mRNA vaccine, in principle, could reach any cell in the body if you run that.
Normally, a virus doesn't come to the heart. That is very usual. And here you have something that can go through the whole system, and if it comes into the blood, it can be pumped around many times and expose all parts of your body, the brain, the heart, all the organs like the liver, everywhere, depending on the individual situation. That means that depending on where this mRNA lands, it can cause a lot of different side effects
in different persons.
And that means it's very difficult to sort of say, this is the correct connection between side effects and so on, because there are so many and they vary. But if you understand the mechanism, how it works, you can tell that depending on where it goes, you have the damage from the immune system.
And from statistics, naturally, you can deduce and get the probability factors for these side effects to occur. And there are such studies nowadays. They are published all the time. But for example, mainstream media in Sweden, there's a total cover-up, or you can't express this.
But the funny thing is that they gave this technology the Nobel Prize.
Yeah.
And for me, this is like one of the most terrible technologies, and has done so much damage, get the Nobel Prize after such a short time.
And it was also, it wasn't the discovery of it was only the Nobel Prize wasn't
It was for the stabilization of the nucleotides
it
in
was
the mRNA.
Yeah, this, I don't know what to say, the uppfinning.
I don't know.
Discovery.
He
said something. What did you say?
Discovery, no?
Yeah,
The discovery.
Yeah, okay, discovery. But well, he Malone, who actually was one of the founder of this vaccine, he discovered himself that it wasn't so good. So he was actually already in 2001 in a state hearing in USA. I observed that he was very much against it, and he talked about the risks about this and how it could transport to different parts of the body, etc., etc. Yeah. So he was, and naturally he wasn't given from the corrupt Swedish authorities or Karolinska Institutet, who is responsible for this. He didn't get the Nobel. It was some Finnish, I think,
some technology development of the technology.
A little bit technology.
My thought on that is that it is, this technology is actually a necessity if you want to vaccinate such a large population, because being used to these technologies that you use traditionally for producing vaccines, it takes too long a time to be able to produce something that fast every year, you know. So they need this technology, and therefore that was more important. Yeah,
totally.
Yeah, that was more important than to be able to vaccinate everybody than the risks, I think. Because I think it is like that. I know it is like that, probably because it's cheaper, it's faster, you can develop much more.
But that's the general discovery of mRNA. But these Finnish guys, they had only did a minor thing, you have said before. The actual prize, it wasn't for this discovery.
No, it was for the stabilization of the nucleotides, actually, the content of the mRNA, because if you put mRNA into a cell, it will degrade quite fast. If you try to express a viral protein in the cell. So this is not, it will not be long enough to be able to...
You know that?
Yeah, I think I know that, because otherwise they wouldn't have done that, because this was probably a failure until they stabilized it.
Okay,
and that is a problem, because you don't know how long is this stable.
Was it Pfizer directed this stabilization process, or?
No, I think it was some other.
Before?
Yeah. Anyway, I think, what was I going to say? I forget now. Anyway, we don't know how long these stabilized mRNA will exist in these cells that are not killed. Can they be there for a year, for half a year, for 2 years? Yeah, but you know, they've shown protein, and that can stay in the membranes of the nerve cells, for example.
I mean, the spike protein.
Yeah, that you can see, but that doesn't mean that the mRNA is there. But still, you don't know. They have not tested before. How long can this stay in the cells? I don't know that, and I think that is something you should find out before you give it to so many people in the world.
For example, we will probably see the death rates in the future.
Yeah,
it will be connected.
I think it's they have made a major crime against humanity, I think, because they exposed so many people, young people, healthy people, for something they really didn't need, to be honest.
Yes,
because
they didn't need it.
They didn't need it for their own health, and it didn't stop the spreading of the virus.
I have actually proved in a mathematical proof that it's suboptimal, just like Ann-Cathrin said in 2019, for these type of viruses, to mass vaccination, the low-risk group, the huge low-risk group. It's simple to do that in mathematics. It's only a principle of proof, you might say.
Yeah.
And also, this is very important, in USA, Martin Kulldorff, the Swedish former
Professor
at Harvard, who was had to resign during the Fauci and the Biden collaboration, let's say. Yeah, they're under their administration. He had to resign because he didn't want to vaccinate himself, because he
already had the infection, and he had an autoimmune disease that could be worsened by vaccination.
The vaccination. Yeah, and yes, and he is now head of Martin Kulldorff is due to after Kennedy has become the health minister there, and Martin Kulldorff now is the head of CDC, and they are
no, he's not head of CDC.
Isn't he?
No, the scientific board of.
Okay, the scientific board of the CDC, but isn't he?
I don't think.
I think CDC is the advisory organ for the FDA to decide
okay.
if vaccines
I'm not really sure, but he has a very important.
Yeah, position. But he is some sort of head there, 15 or 18 people or so, and well, we hope that he will. And you say they are. I mean, they are totally against it now in the.
Yeah, he understands that it's not. You should test things before you.
Yeah.
Really in a depth before you give it to so many people, healthy children, healthy young people, and you should not sort of experiment with people. And I think that is good. He's not against vaccines in general, but to expose people for something that's new and not tested.
And there's one thing we haven't talked about already, and that is the vaccination of children, who Ann-Cathrin has talked about a bit. But it's in these years, it's the younger years, and when the thymus gland in younger persons, they are having a mechanism, selection process, and one of these two processes, and one process is to determine if T cells are going to attack and have a memory of foreign proteins, and if
it's actually making it's difficult. It's teaching the T cells to react to foreign protein and not to self protein, and also to bind properly to the right cells.
And if they vaccine a lot of such young person and children, there's a risk for that they there's a risk of that they tolerate a great deal.
That's one thing. Another risk is the increase of autoimmune diseases.
And both.
Yeah. But that is also something.
And they were very. This is one interesting discovery, I must say. In Uppsala region, they found that there were a lot of vaccinated people against the testing.
TB.
TB, the TB virus. And
encephalitis.
Yeah, and they were actually everyone, I mean, nearly everyone who had this, who had
tick, I think it's called tick encephalitis.
Oh,
no, sorry.
Okay, but they...
Is it Lyme?
They found that
disease?
It didn't function.
What did you say?
TBE, is that Lyme disease in English? No?
Maybe.
Yeah, I think so, Lyme disease.
Yeah, okay, thank you.
But and they found that they were, even if they were vaccinated, these people had the infection, so it was non-effective, and that might be associated to?
Yeah, it can be that these people have received, because so many people has the Lyme disease vaccinations from the tick-induced encephalitis, that these viruses have mutated somehow. I don't know.
Yeah, but also that if they are vaccinated as children or younger people, it might be due to this interference with the thymus gland process. Yeah. So in a natural environment where you get these foreign types of non-pathogenic proteins, etc., from then this process seems logical. It would even children. So I think it's connected to the to when the environment for children, for example, gets too clean, then it's not. That's not.
That's actually a reason for increasing
allergies. Yes.
So my perspective is that you shouldn't over-vaccinate children because they still develop their immune system,
including the thymus. The process.
Yeah, the process is important, and that is part of the development of the immune system. So I think you can give some vaccines, of course, that for very severe diseases, but you shouldn't vaccinate them for influenza or flus or colds or things like that. Because
if the virus connected to the vaccine, if they start to mutate,
of course,
you have to know that.
Yeah, of course. What did you say?
I just want to ask you. So you say that you shouldn't vaccinate children. You shouldn't over-vaccinate children.
No.
So I don't know which vaccines are on the children's schedule here in Sweden.
Yeah.
Do you have an example of some vaccines that maybe shouldn't be on there?
No, my criticism towards this type of vaccination is that you vaccinate towards too many things at the same time.
Also.
And that is not good, because that increases the risk for development of autoimmune diseases, because you have too many targets they should react to. And also you don't have it might be that you don't have as effective protection against all of them if you do it at the same time, because some could be more effective and then they will dominate the response towards this vaccine. So you might still be not protected, have a good protection.
You must say that it's too much trial and error, and the errors seem to be.
I think what they want to say, give one injection and it's over with. But as an immunologist, I don't like the idea that putting together too many vaccines and give it at the same time. And another thing I have criticism against is that you give it too early. You give small small children vaccines. And to my knowledge, you shouldn't give vaccine until after you are 2 years old, because then is where it's really have an effect, because your immune system is not developed enough to be a good result. And your mother, if you give, I don't know, if the mother feeds the baby is protected by the mother's immune system through her milk, because the antibody is there, and the baby will have the same protection as the mother. So I think the reason for that is that the immune system in the baby is not developed properly to that vaccination before 2 years old is a good idea generally, because but I don't know.
But you can see
that is something I think you should analyze.
Yeah. You can see in the USA now, Canada is known for in mainstream media to be a total anti-vaxxer, but there are some new evidence now from studies, etc., that it actually is very complicated with these mass vaccinations for children, for example, because in USA they have been vaccinating for a long time, and in this I would like to recite an older study regarding generally, not only for children, but when they in USA they increased the vaccination rate against seasonal flu in the 1980s, middle of 1980s, from 30% to 67% about 10 years later. What happened about seasonal flu and death rate? Well, it didn't decrease. It actually increased during that time. And then during that time, there were no pandemic or etc. So it was an early observation that indicated there's something wrong with this theory about seasonal flu vaccine, even among the elderly. Yeah. So, and it's naturally the most logical thing is that very many elderly, they have had a seasonal flu before, or a pandemic like Hong Kong or in Asia before also that. So it's unnecessary for most part of these elderly to vaccinate, because what can happen? During a pandemic in the wintertime, it could be this unbalanced effect that actually increases their risk for death. So it's totally, they are not following good science here,
no,
I think.
I think that you are actually you don't get your T cell memory updated, because we are always surrounded by viruses or influenza virus every season. But
which are good for us.
Which are good. Most of them we don't even feel sick, or we have very mild kind of cold. And that is due to that, we still update our T cell immunity to the latest version. But if you give your influenza shot every year, you don't get this update. And then you have 10 years or 15 years or whatever, how many years you have given that
even
vaccination.
If it's updated.
Yeah, you don't get updated. And then when you get the next, when you get infected, and the vaccine doesn't protect you, you can get really sick, because it's like you have to start from an earlier point, because the virus has developed, but your T cell immunity hasn't during this time. And that means that you can get more sick from this.
And you can relate that Canadian study.
Yeah, the Canadian study showed actually during the swine flu pandemics, there was a Canadian research group that found that people who had every year vaccinated against the seasonal flu had a higher death rate from
swine flu
than people who didn't get the seasonal flu vaccinations. And this could be the explanation that they didn't update the broad T cell immunity. And actually what is called asymptomatic infection is when you get exposed, you get infected, but you don't get so much symptoms, because the infection the virus is removed so far.
And that is what we talked about before, the Diamond Cruise, Diamond Princess ship.
Cruise ship,
yeah. And with all those asymptomatic infected.
Yeah,
elderly.
And so that's if important. You have this, you can sort of protect yourself. But if you are so weak in your body that you cannot handle an infection, then it's actually could be worthwhile to protect you with an seasonal influenza shot, because in that case you might not survive and you can survive another year, for example. But in that case, it's special. But if you are healthy, you are young and so on, you don't need to get these shots.
This doesn't actually mean that we are against, for example, vaccination against the bacteria infection. Like, for example, during the last Second World War, I think that type of vaccine.
Or not against polio vaccines or smittkoppor.
No, but we don't. I don't like the polio vaccine because it's actually that I realize now that it's probably the reason.
For your problems,
Yeah, but an injury affecting my running muscle isn't functioning as optimal as it could be.
Yeah, good.
Yeah, there's some I've heard from Joe Rogan. Just he has some guests that are against the polio vaccine. I have not done my own research on it, but they argue that most polio nowadays are asymptomatic from the polio.
In my case, it was the type of polio vaccine. It was a weakened polio vaccine. So it was the virus itself. And of course, there's a certain risk with that. And because it's if it functions, it has entered into other infection cells. Otherwise, it wouldn't function.
No, but it can go into your nerve cells.
Yeah, and
the nerves.
Yeah, and that's my suspicion in my case. So,
but anyway,
there are different types of vaccines.
Yeah, there are different types of vaccines. But there's a scandal connected to the polio vaccine that was one of Bill Gates.
Recently
in Africa, where they gave oral vaccines to young children. And that was living bacteria. And you have immune system also in your intestine. So small babies and young children got. Yeah, it was bacteria. It was it was polio virus. It was virus.
Yeah.
So it contained live virus, and it was given orally to the children as a shot or something like that. They gave it like that, and the problem was that this was living bacteria, polio bacteria, and they went through the intestine. So when the grown-ups around these children were changing the diapers or whatever, they were exposed to this virus. So it actually caused a lot of polio infections secondary to people from the feces of the baby. And that is what is called shredding of vaccine. So that a vaccine can infect others because it was live living bacteria.
Okay.
Oh, virus, sorry. It was functional virus, sorry.
What's something I must now also that one basic type of cheating, and the first one my type of discovery in Israel, I think it was, I've never heard it, seen it before, and it was after that we were getting suspicious of the vaccine studies and the vaccine effect studies, and it was in those days COVID-19 that was we found that I had found, and they published. Ann-Cathrin was together with Läkaruppropet, the doctors of Sweden, many doctors of Sweden who were protesting against this because all these unscientifically studies, and they analyzed the Swedish statistics of COVID-19 deaths from vaccinated and unvaccinated, and they discovered, of course, that it was a lot of cheating there. They had a lot of people who were so-called unvaccinated were in actually they had got one shot, one vaccination, or even up to 2 weeks or 2 or 3 weeks. It's different in different statistics and Swedish. And they, the Folkhälsomyndigheten, the Swedish authority, they published their effect studies or the effect statistics, you might call, and Folkhälsomyndigheten, then it was so false regarding this. You can call it selection bias or so, but it's a general type of cheating where they call a lot of vaccinated for unvaccinated, and they are included in the results for the unvaccinated group. And this is during a pandemic. There is obviously this higher risk for getting this unbalance effect when you infect, you are higher risk of getting infected in the similar time, same time, more or less the same time as you are vaccinated. So, and these times are, as in the Israeli, these high risk times are eliminated, and they were actually included in the non-vaccinated group results, these deaths, for example, and they showed that in exactly, it was during the year 2021, the whole year, it was 135, or I think it was 135% more deaths among the vaccinated than they had said. So it was a huge.
And this was in the group that has not, by their inclusion definition, is vaccinated. That means people who got shot but didn't have got enough time before they calculated.
But they were in very high, as I said.
There were very lot of people in that group.
So and it's partly when you mass vaccinate during a pandemic,
it's crazy.
It's totally loco, as they say in Spain, and what happened then after I had contacted this authority in Swedish, Folkhälsomyndigheten, in several mails, and we had warned them against this effect, and warned them against from this Israeli results or disaster, and also had warned the Läkemedelsverket, the authority responsible.
It's the same as the FDA, I guess.
Yeah, correspondence to FDA in USA, corresponding authority, and we warned them against this, and this explicitly this disaster in Israel, and they didn't bother. But what they did bother, they saw that I had used one of the diagrams from the Folkhälsomyndigheten itself. It was actually an evidence against them, and they had not understood that before. But after that, I've used that and mailed it to them. They unpublished, they censored this diagram. Their own diagram. But they listened something, because afterwards they have in the Folkhälsomyndigheten, they say nowadays that but you should not be you should not be infected when you vaccinate, because it might be good for the immune system to only concentrate on one thing.
But what is funny is now the Folkhälsomyndigheten recommend people to get the vaccine that is not really working against the new variant of influenza virus. That is really crazy.
Yeah, and that's
then you have no use, but only the risk of getting infected.
Yeah. And we have sent new mails about this even to the government and to all these responsible authorities, or who should react if they have some sort of responsibility in their minds. Even even certain, I mean, the individual people should understand. They should have some problems with the conscience if they are doing this. And
Yeah.
so
anyway, you shouldn't really recommend people to take that, because if you have an ineffective vaccine, it's just going to disturb your immune system. And
you add risk.
You add risk, but you have no.
But they say that it's this lie they have, unscientifically proved lie about protection against severe disease and death, and that's it's not possible. It's against the whole principle of vaccine.
The only thing, if you can really stop the infection, that is what antibody can do if you get it in time. But if it doesn't work, what is the use? Nothing.
It's only a higher risk.
Yeah, you have to. That's the idea.
But they are probably somewhat in all these deals with EU and the vaccine both companies, etc., etc. So we hope that the American
policies.
Now they have changed. They are thinking about a little bit more scientifically now again. Regarding all these things. So
but I think this is a European Union strategy, not to try to prevent all kind of diseases with medication, vaccines, so that people won't need too much treatment.
But it doesn't function.
It doesn't. It's not going to
this
work.
Type of diseases like common colds and influenza and even
but all
related
kinds of diseases.
They
try.
But these that are not possible to do.
But I think it's also with other drugs that you should treat this, you should keep your blood pressure in that level, and so on, and you should blood fat in that level, and the blood sugar in that level. Okay, but you overtreat people, and all this medication has side effects, and then you get problems from the side effects. So you treat more people to save a few, and I think that and I think the same with vaccines. It's the same logics, but it will not work because it's always a matter of balance, and some people will have side effects and will not benefit at all from this.
And regarding, for example, the COVID-19 pandemic, why didn't they think about it? Why has it taken 3, 4, 5 years for this to end? And when Hong Kong influenza, when they didn't have a lot of vaccines and they didn't have as good hospitals, why was it ending just in a few years and with far less deaths from per capita in when they didn't have these high level hospitals, etc., for protecting the most sick, etc. But they were protecting. Influenza is regarded as more severe than common colds that COVID-19 is related to. Also, the one thing is probably different, we think, and it's due to this gain of function research, and we think that there might be, they have found actually in the vaccine, and also probably therefore in the COVID-19, in the SARS-CoV-2 also certain DNA sequences or RNA sequences in the virus that are actually human
derived.
Derived from humans. So it might be connected. And my theory is that in some way it's connected to these They are found in studies that even COVID-19 infection can, and it might be connected to this long COVID, you say, they can reduce the effects of actually
mitochondria.
The mitochondria might be, they are shown to be some problems with mitochondria after the vaccine, after even after the COVID-19 itself. And I think these founds of DNA and RNA, etc., in the virus, RNA in the virus, but that there's some connection there. And I also experienced myself that I have trouble long term nowadays with some sort of problems in the throat that I never had before, and also some since I'm a runner, I was also some difficulties with I think feeling of lactic acid in running. I hadn't had. That was after I had COVID-19 that problem
occurred.
Yeah, occurred the first time. And when I heard about these founds in these founds in USA in that hearing, it was about also these mitochondria sequences. So in the vaccine, so it's something. It's more severe than usual chemical, but that doesn't mean that you should vaccinate it, or that hasn't that's no logic for that. It's the same principle against this mass vaccination as before.
Yeah, it's a lot of information to take in, but I feel like this is the perfect media format to do things because I feel like when you watch television and traditional media, you get like short segments when you can elaborate on your point, but we have sat here for like 2.5 hours soon, and you guys have really elaborated on these issues, and I feel like it's been very interesting to hear from you guys, and I feel like this alternative media thing that's going on right now with podcasts and things like that, I feel like that's the future, and these long format conversations are what people really want to hear, to really dig deep and dive deep in these kinds of subjects, because they are nuanced. It's not black and white.
It's all, there's a bias effect there also. I think it's those who are already critical and understand maybe that takes their time to listen to get more angles about it. But there are a lot of people that say, "Oh, you are," and they are so changed in the mind, so they don't listen.
The discussion because they don't want to get their world disrupted, because they have these ideas, and it's like a religion. If someone say, "I don't believe," and you have to listen to this, you just shut down, and that is normal. But I think also that, from my perspective, I try to give very, in my Facebook and so on, I have tried to get very detailed information, and that is because I want to give people arguments for themselves. The people who are really interested, they know, and they can spread this information further, and that is, I think, what is important. Even though you have people who are pro what we are saying, and then they understand what we are saying, they can actually get better arguments, because they are scientific arguments they can put forward to people who are criticizing them for not wanting to take a vaccine for a flu or whatever. They have the arguments, and that might strengthen them in this.
And I want to add something there, and that's the main principle we are talking about when regarding the well done vaccination against these types of common colds, etc. No, common cold is another thing. It's very difficult. But the influenza, for example, the main principle here, the interaction, that it's not only antibodies but also very, or more important, long term, the T cell response, and therefore I have a corresponding. All my analysis, it's several hundreds. Very usually you can discover, but they don't know about the broad T cell response against the conserved inner proteins. And that principle is the most important principle to understand. And it's also most important to understand that it's their spreading that's causing the flock immunity. Herd immunity in English, yes. And it's not the vaccine, it's not the antibodies in general, especially not if they have vaccinated with a primarily effective because then there are these new variants that are caused by this mass vaccination, and you get a longer and less effective vaccine, etc., etc. So what ended the Hong Kong influenza and in Asia in 1940s, it was the broad T-cell response against that ended if there were any variants. If there wasn't any variants, you also had a big use for that in the future against future pandemics, as we saw in the swine flu, for example. But in those days, the antibodies may have functioned better because there wasn't so many variants, because they didn't mass vaccinate. But
what I would say is what you have to understand is there's a balance. There's a balance between virus development and herd immunity. And the more you vaccinate, the more you drive for virus development. So what you are doing...
You take time and take more time.
What you do, you're speeding up the virus development by mass vaccinations in these cases. Because otherwise, you are putting an evolutionary pressure for the virus that are mutated, that can pass by the antibody response and effect. They have an advantage over those that the vaccine antibodies can attack. So you will actually increase the number of mutations, and also it will go much faster, just as we have seen here with the COVID pandemics. You have lots of new mutants coming after each other, and this is a typical result from the mass vaccination, but also from isolating population from each other. So actually what you have done, you have made things worse by these interventions.
As they did with the lockdowns, for example.
It's a combination. They have locked down.
Regarding the lockdowns, I must actually add that even in mainstream media, in Swedish television, they had in the scientific related program, they stated that researchers had showed that about 2 million people or 2 million childs in less developed countries had died due to these restrictions regarding and since they were also in the gross national products were severely affected in the whole world regarding transporting foods to children, etc., in Africa or other countries. So 2 million extra deaths due to these lockdowns. So
and that was a total experiment, because nobody had experience from that. It's just someone's theory.
It could be deduced before.
But it was someone who thought it was a good idea, but they have never tested it, and they don't know what they are doing.
No.
If you understand both how this virus work and how immune system work, you could immediately say this is not effective.
And regarding one more thing about media coverage, etc., and cover up and non-cover up, in 2019, you asked before about the media then. It was actually in about a few months after this, it had declined or so. Anyway, it was after the acute phases and so forth. And it was one paper, Dagens Nyheter, at that time, they had a scientific reporter who said she did talk about or wrote about the 4 different types of critics, and she thought that there was one that she actually thought was probably in her mind was the one most interesting or the best one, and it was the pair Engwans she talked about, and they had something there. She didn't probably understand why, because she wasn't educated at all. Like there are now many scientific reporters who don't know what they're talking about.
In general, they are not specific in the way that.
But at that time, we were at least not anti-vaccination, in her mind.
But now, if you criticize, you are an anti-vaxxer. That doesn't matter if you are sort of coming with scientifically correct criticism. You are still an anti-vaxxer if you just have questions about the vaccines.
But we are now, as I've stated before, we are now acting against the authorities and writing mails with all these references to what we have discovered, etc. Yeah. And in a few times also maybe different type of scientific studies that are supporting our conclusions, and so we are not giving up. So I mean, it's interesting that I think they have proposed a law or something that some type of criticism of even people in authorities, it should be illegal. Yeah. So they can actually, I mean, act unscientifically. If they do that, maybe you wouldn't be able to even criticize them.
That is crazy.
And that it's been some cases in Europe. I think in Britain there some these type of criticism. It's one who is in jail for this, and also in Germany, I think one researcher that or something like that, who is put to jail. So, but what does it mean to do that? Put one person in jail, and that's only triggering those who are criticizing is to understand that they are totally loco.
Yeah, the problem is that you have a lot of problems with criminality, and you put your focus to arrest people who are coming with the criticism.
And now we're trying to save people with their criticism.
This is a very scary sign of a kind of a dictatorial system where people are not allowed. You have to just obey, and I think that is really scary to me.
I have already
I just wanted to say I don't think they have free speech in Britain anymore, because this year they have had like 12,000 people have been arrested for media posts like on Facebook or on Twitter or
crazy.
It's crazy.
What type of government is it now in England?
Socialist, isn't it? Or Labour?
Keir Starmer is
it's a left. Okay, I think it's typical of left wing, but maybe we shouldn't talk about that because
that's a whole other discussion.
Then 50% of the listeners think we are anti-vaxxers only.
But I think it is like it's a different level. It's actually, I think the politicians are just puppets, and there's some higher general decision made that should be valid in all the states, like the United Nations, WHO, for example.
The politicians themselves doesn't exactly know what they are.
No, they are just puppets in many cases. They follow the regulations, the rules. We have to follow the international regulations, blah blah blah. So they are actually locked in. I think that is who is really making the decisions.
it's many different organisms. It's a very complex. It's so what will happen is not you can't deduce exactly, but it seems impossible to cover up all these injuries. I have a last observation I will relate, and it's you can see on the streets, for example, here in Fuengirola or even in Swedish big cities, I've noticed that a lot of people, elderly, maybe 50 years or older, a high percentage, maybe between 20 to 40, when they walk more like a duck nowadays. They tilt like this. It's a little bit like when my injury in only my left hip from this probably this polio vaccine originally. I don't walk like that, and I can run, but I have trouble in high speeds, etc. But they walk like ducks, and it wasn't before, and probably now people are getting used to it. There are also many vaccinated.
Maybe they're getting some nerve damage.
You can elaborate on that.
Yeah, maybe they get some nerve irritation, problems with the nerves.
How does the virus reach, or the mRNA reach?
Through the blood, and it can go everywhere.
But how can they? I mean, it's probably nerves. Is it regulated? Is it stimulated from the brain, or is it low?
Could be both, but I think it's local.
It's very. They are not.
If it's depending on I think
it's symmetric. It's symmetric. Both legs. So it should be up here something.
Can be up here, or they can be back there.
Okay.
Yeah.
So it's very difficult. You have to examine them to tell.
And there should be studies now. Have been.
Yes.
If
there
you
should
Google
be many studies.
Maybe there are a lot already.
Yeah.
I haven't Googled on that thing, but it's so obvious.
Yeah, and the fight will continue. Guys, we need to wrap this up.
Yeah.
Any last words? Where can the people who have listened to this find you guys for more information? And
we are writing at our Facebook's
account.
Yeah, and my homepage contains some of the proof of things I have found, like the Israeli case and so on, with some links, and also some links about several. Ann-Cathrin has so many interviews. I haven't systematically all these links, but she has some very good interviews where she talks like this, but even more stringent, I think.
More scientific, maybe.
Yeah, because she was interviewed, for example, a pod that was very high rated before recently about I think it was some kind of nurse who interviewed you, and that was strictly scientific and not so much other political associated discussion. So it was, and it was not all, but it was so Ann-Cathrin was in top form then, and we have both been in good shape today also, I think.
Yeah, you've been. Yeah, I've really enjoyed listening to you guys. It's been learned a lot, and I hope the people who has been watching this also have learned a lot and have some arguments if people call them anti-vaxxers in the future, that they could.
That's important.
Yeah.
To strengthen the people who feel it's wrong, but they don't have the knowledge.
Okay, how much will you censor of this 2.5 hours?
Nothing. I will put it up raw.
Okay, then you have to wake them up so they can endure. Maybe you should before say that they should exercise a lot to get your brain a bit more efficient, and maybe you can stand these
this last one you will
2.5 hours.
Who says good talking?
Okay, Ann-Cathrin and Bo, you're welcome back anytime you want. So
thank you.
Have a good time, and we will see you guys next week.